Hello everyone, how are you doing? I would highly appreciate this opportunity in order to talk to you in this marvelous morning. My name is Khalid and this is my first video for those who are interested in field of molecular genetics or molecular biology. Today I'm going to talk about something called the DNA extraction. The DNA extraction. So, what is a DNA extraction? The DNA extraction is an isolation process of the DNA from the other cell components, such as proteins and other organelles. What is the DNA? The DNA is a stand for the deoxyribonucleic acid, and as you know that the DNA found in the cell and within the cell found exactly in the nucleus. Am I right? So the DNA extractions, the aim is to get the DNA. First, we have to break the cell membrane, remove the proteins and other contaminants in order to get the pure DNA. Right? So the DNA isolation application. The DNA isolation application used in many fields such as scientific use in the introduction of the cell into the plant or animals or human beings such as gene cloning, and used for also for forensic science such as the determination of the paternity or the rabies or thieves or victim wars. So in order to isolation the DNA, there is a things we have to do firstly. So I'm going to talk a little bit about the sample or how to collect your sample. I'm talking here about the whole blood. So the samples of DNA can be from the blood, from the tissues, can be from the bones. So the age of the sample can be fresh or can be frozen. So methods of DNA, method of DNA isolation. There is many methods. Usually people use the traditional methods such as organic extraction and salting out. I'm going to take a little bit about the organic extractions. In the organic extraction method, people use phenol chloroform. So when you mix the phenol chloroform with the cell list, two faces occur, or you're gonna see the two faces. The upper face is the DNA and the lower face is a phenol. So you going to remove the DNA. Also, the salting out methods at the higher cell concentration, the DNA is hydrated and precipitated and low solubility. So when you use the high salt concentrations such as ammonium acetate, sodium chloride, the DNA is dehydrated and low solubility and then gonna be participated. So I'm going to remove the DNA and remove the protein and other contaminants. I myself would prefer to use the kits in order to do extraction process. One of my favorite kit is Interon, as you see here in the picture, Interon kits. So this is I think this is the safety and give you accurate results than the other two methods which I mentioned earlier. I'm going to stop here. Thank you for watching. See you later. Bye bye.